very much for the introduction and for organizing this seminar for me. And also thank for everyone who took your time to listen to my talk today. So today, I will talk about my recent work on characterizing the single nucleus transcriptomes of the skeletal muscle cells, which I believe that opened a lot of new exciting possibilities for the field. So as everyone in this audience knows, the cells are the basic building blocks of all life forms. And also each of you might have your favorite suborganelle in the cell. I believe that everyone will inarguably agree that the nucleus is the most important organelle in any given cell type, because the nucleus stores the DNA and also regulates the gene expression, which confers identity to any given cell type. Now, when you look at the cells in a nucleocentric manner, you can roughly classify 4 types of cells to 2 categories. The first category is mononuclear cells, which have 1 nucleus in 1 cytoplasm. And this is a case for most of the cells in our body. Now, the second category is multinuclear cells that have more than 2 nuclei in 1 shared cytoplasm. And in our body, there are a few types of cells that have this kind of architecture, such as skeletal muscle cells, placenta cells, and liver hepatocytes, as well as heart cardiomyocytes. However, before going to the main part of my talk, I just want to give you one definition, my own personal definition about syncytial cells. So whenever I say syncytial cells, I want to make it clear that I'm not equalizing the term syncytial cells to multinuclear cells. So what defines syncytial cells? So please take this with some grain of salt, because this is purely my personal classification, and this is not something that appears in a textbook or Wikipedia. So in our body, there are basically 3 types of natural true syncytial cells, which are skeletal muscle cells, or osteoclast cells, which remodels our bone, or some types of placenta syncytial trophoblast cells, which supports embryogenesis. Now, other types of multinuclear cells, but are not true syncytial cells, are, for example, liver hepatocytes or heart cardiomyocytes. Now, in terms of their number of nuclei, generally, true syncytial cells tend to have very many nuclei in 1 cytoplasm. Even for very small size osteoclasts, they easily have 10 nuclei per cell. And in case of big myofibers or placenta cells, they can have hundreds of nuclei in 1 cytoplasm. Whereas in case of these other multinucleated cells, they only have 3 to 4 nuclei maximum per cell. Now, in terms of developmental mechanisms, the true syncytial cells always formed by fusion of mononuclear cells, whereas these other multinucleated cells develop through endoreplication, meaning that these cells undergo cell cycle without finishing cytokinases. Finally, in terms of functionality, these true syncytial cells need to, the formation of the syncytium is essential for these cells to function. So if the skeletal muscle cells cannot multinucleated, then the contraction will be completely lost. If osteoclasts cannot be multinucleated, then they cannot remodel the bone. And if the placenta fails to form syncytium, then the embryo will die. Whereas in case of these other multinuclear cells, actually the mutants that blocks their multinucleation does not seem to show any obvious phenotype. And at least for the heart cardiomyocytes, it has been shown that the multinucleation of cardiomyocytes is disadvantageous for heart regeneration. So whenever I say syncytial cells, it refers to this type of cells which have many nuclei in 1 cytoplasm, developed by fusion of mononuclear cells, and their syncytium formation is essential for their biological functions. So coming back to the comparison of mononuclear cells and syncytial cells, due to their unique cell anatomy, these syncytial cells face a number of challenges that regular mononuclear cells never have to deal with. For example, unlike the mononuclear cells, these syncytial cells have to regulate gene expression programs coming from many of their nuclei. Now, the syncytial cells also have to really regulate how far local signaling pathways would travel throughout the cytoplasm and how that will determine the cell fate. So, for example, if there are locally present growth factors or stress cues, the cells have to somehow control how far these signals will travel in the cytoplasm, and depending on that, it will either act very locally in the cell or alter the entire cell fate. Now, syncytial cells also face a unique problem of nuclear homeostasis. So in mononuclear cells, when you have a damage on the nucleus or on the DNA, there are basically two choices for these cells. They either repair this damage, or if their repair capacity cannot catch up, then these cells will undergo cell death. However, in the context of syncytium, it will not make much sense to kill the entire cell only because a subset of their many nuclei were damaged. And alternatively, these cells can adopt another choice, which is to remove this damaged nuclei, which is completely ununderstood process in biology. And eventually, this missing nuclei must be somehow activate locally present stem or progenitor cells, which then recruit these cells to the syncytium and make them donate and regenerate this missing nuclei. So I can go on this type of list for another 5 minutes, but of course I will not do that today. But the real bottom line of all these examples is that syncytial cells really present very unique and exciting paradigm at the interface of cell biology and developmental biology. So my research uses skeletal muscle cells as a main paradigm to study this unique biological properties associated with the syncytial cells. So when you think about the skeletal muscle cells, you might think that it's quite boring or very boring organ because all it does is to generate the force and make our body move. But I want to emphasize that the skeletal muscle is way more than and much more than just inducing contraction. And actually, the skeletal muscle is highly involved in regulating whole body metabolism. Now, the skeletal muscle is a well-appreciated endocrine organ. And throughout our lifetime, the skeletal muscle has to support tissue regeneration as well. Therefore, a key question regarding the muscle function is to understand how these cells can coordinate its so many functions at the same time. And this is probably executed by the fact that although these cells are highly multinucleated, each of their nuclei are not identical, but they are interfacing different types of microenvironment. For example, some of these nuclei are in close contact with the motor neurons, whereas others are facing, for example, the tendon, mesenchymal cells, blood vessels, immune cells, and so on. And therefore, it is possible that these different cell and nucleus interactions guide local gene expression inside a muscle, and that this plays an important role in orchestrating many functions of the muscle cells. Now, indeed, previous studies have identified at least one transcriptionally specialized nuclear subtype in the muscle, which is located at the junction between the motor neurons and the myofibers. So what happens in this part of the muscle is that the motor neurons send signals such as A-green, which are perceived by specific receptors expressed on the myofiber. And this leads to downstream signaling pathways, which induce the expression of genes that are required for neurotransmission. And as a result, muscle contraction can begin from the very center part of the myofiber. However, other than the neuromuscular junction nuclei, it remains unclear what kind of nuclear subtypes actually exist inside a muscle cell. Now, another interesting aspect of the muscle cells is muscle nuclei is that their locations can dynamically change in different conditions. So in the normal muscle, their nuclei are located at the very periphery of these cells. However, during regeneration or in a subset of human myopathies, these nuclei gets relocated to the center of the cells. And although this phenomenon have been known for a very long time, we still do not know whether and how the transcriptional programs in these nuclei are different from the others, and more importantly, how they contribute to muscle regeneration or disease progression. So the obvious first thing to do in this line of research was first to know what kinds of nuclear subtypes actually exist inside a muscle cell. So as I mentioned earlier, around half of the nuclei in the muscle tissue actually are not coming from the multinuclear myofiber, but they are derived from these diverse mononuclear cells that surround the myofiber. So we started this by genetically labeling only muscle nuclei with histone to be GFP reporter. And this was possible by crossing HSA Cree driver, which is specific only in the syncytial myofiber cells, which would recombine and drive the expression of histone to be GFP. And as you can see here, we were able to confirm robust labeling of the muscle nuclei in uninjured muscles, as well as when at early time point of regeneration after we gave injury to the muscle. So to this end, we collected around 5,000 muscle nuclei across different time points of muscle regeneration. And as a side note, in this injury model, using cardiotoxin injection, regeneration is completed around by 3 weeks. And one myofiber typically contains around 200 nuclei, which calculates to about 5 to 10 myofibers at different time points. Now, in regards to the sequencing platform, we actually used, we didn't use the most modern technology, the 10x Chromium, but we instead used CellSeq2, which is lower throughput, but still has higher gene detection sensitivity. So this is how it looks. Shown on the left is the UMAP cluster plot indicated with different nucleus subpopulations. And shown on the right is the same UMAP plot, but indicated with different time points. And for those in the audience who are not familiar with this type of graph, basically one dot represents the average gene expression profile of single nucleus. And if two nuclei are close, if two dots are close to each other, then their transcriptomes are more or less similar. And if they are far away, then their transcriptomes are very different. So as you can see here, the majority of the nuclei were clustered in the center of the UMAP plot, which we named as Burke myonuclei. And as you can expect, the genes that are specifically expressed in this Burke myonuclei were mainly involved in the basic functions of the muscle cells. And the exact list of the genes actually differ depending on which time point within Burke myonuclei you are looking at. So if we mainly focus on the uninjured muscle, these genes are mainly involved in controlling metabolic pathways, or those actually are known to inhibit muscle growth. Now, if you focus at the early time point of regeneration, you can find genes that have been implicated in active muscle growth, such as IGF-1. And this makes a lot of sense because at this time point, the muscles need to grow back. Now, if you focus on late time point of regeneration, many of these marker genes were involved in muscle contraction, indicating that the overall muscle function is getting regained at this time. But more importantly, we identified a number of nuclear subtypes that had very distinct transcriptional profiles, which can be easily appreciated by the heat map on the right side. And here you can really see that each of these clusters is defined by expression of hundreds of different genes. Now, for these experiments, we used the TA muscle, which is one of the hind limb muscle in the hind limb of the mice. And in the TA muscle, there are 3 types of myofibers, which can be classified based on the specific expression of different myosin isotypes. So one possible explanation was that whatever heterogeneity I'm looking at is just due to fiber level heterogeneity, not due to real heterogeneity at the nuclear level. But when we look at the expression profile of these myosins were evenly distributed across all these distinct nuclear subtypes, indicating that this heterogeneity is not explained by fiber type differences. So the first thing we tried to identify from our data set was nuclei at the neuromuscular junction, because this will really ensure that the entire approach is working properly. And very gratifyingly, we are able to clearly identify nuclei at the NMJ, which are characterized by specific expression of a number of known NMJ marker genes, such as PK regulatory subunits, acetylcholine receptor subunits, as well as acetylcholine esterase. In addition, we identified a number of novel NMJ genes that were not known before. And when we performed double in situ hybridization using PK regulatory subunit in red, which is one of the known NMJ genes, together with a couple of newly identified NMJ candidates, we were able to see robust co-localization of these transcripts, confirming that these novel candidates are indeed expressed from the neuromuscular junction. So after we confirmed the presence of neuromuscular junction nuclei, then we asked whether we can identify myotendinous junction nuclei. So the myotendinous junction is located at the very end part of the myofiber, where the myofibers attach to the tendon. And the main role of the myotendinous junction is to buffer the mechanical force applied to the muscle tissue. And for that reason, the MTJ is a major site of injury when we play sports, and therefore have a huge implication in muscle rehabilitation and regeneration. However, despite its well-appreciated functions, it remains unclear whether these MTJ myonuclei are really transcriptionally specialized. And quite interestingly, we could identify two nuclear subtypes that express genes related to the myotendinous junction, which we named as MTJ-A and MTJ-B. And in case of MTJ-A, these nuclei were characterized by specific expression of genes such as TIGD4 and collagen 22A1. And especially this collagen 22A1 has been functionally characterized in the zebrafish model using morpholino knockdown, which completely disrupted the myotendinous junction development. Now, in case of MTJ-B, these nuclei express alternative set of collagens, which were all known to be deposited at the myotendinous junction. And especially the deposition of this collagen 6A1 was notable because mutation in this specific collagen is known to cause Best-Lin myopathy in humans, which is characterized by specific deficit at the joint part of the muscles. So the further characterization showed that MTJ-A nuclei represent the myotendinous junction nuclei that are present in all myofibers. So what you are looking at here is the longitudinal section of the entire TA muscle. And on the bottom of this image is where the tendon is located at. And in this boxed area, you can see that the tendon is penetrating into the muscle and forming the myotendinous junction. Now, if you look at the in situ hybridization for TIGD4 on the right, you can really see that TIGD4 is expressed only at the MTJ area in the entire muscle, basically decorating throughout the myotendinous junction area. Now, when we zoomed into this image to confirm the expression of TIGD4 from muscle nuclei, that was indeed the case. So in these images, on the left side are the tendon, and to the right side of the tendon, you can see these arrays of myofibers coming and contacting the tendon. And when we performed double in situ for TIGD4 and collagen 22A1, the two markers of MTJ-A, you can see that these transcripts are co-expressed in these nuclei, which are GFP positive and located exactly at the junction between the tendon and the myofibers. Now, when we look at the late time point of regeneration, their expressions became much more pronounced, indicating that the myotendinous junction is actively getting regenerated at this stage too. So to definitely demonstrate transcription heterogeneity in single situ, we also isolated single myofiber and performed double in situ hybridization. And here you can see that UFSP1, which is one of the newly identified NMJ genes in this study, is expressed only from the middle part of the myofiber. And at the same time, TIGD4, the marker of the myotendinous junction nuclei, is expressed only from the very end parts of this isolated myofiber. So I just want to briefly mention that MTJ-B nuclei were also validated. So we could identify GFP positive nuclei around the MTJ, which co-express marker genes such as PDGF receptor beta or collagen 6A3. However, unlike the MTJ-A nuclei, which were present in all myofibers, MTJ-B nuclei were found in only subset of myofibers. And for that reason, for some time, we were really not sure about what this MTJ-B meant in a biological point of view. And we are really delighted to see back-to-back papers from Delphine Duprez group in Paris and Pelek Hassan's group in Israel, who together showed that during development, there is a specialized subset of fibroblasts that selectively fuse to the end part of the developing myofiber. And this nuclei in the future becomes the myotendinous junction nuclei. And based on that, we currently think that this MTJ-B population might represent developing or transitional myotendinous junction nuclei. And probably in agreement with that idea, we could see that there's a considerable increase in the proportion of MTJ-B nuclei at late time point of regeneration. So far, I have shown you that we could identify the previously known population at the NMJ and the expected but never characterized populations at the NTJ. Now the rest represent completely novel nuclear subtypes, and I will not go into the detail today, but I will just give you the time markers and give you the speculator function of this rest of the nuclei. So in case of the first novel nuclear subtype, a lot of their marker genes appear to be involved in trafficking at the ER and Golgi domains. And in case of the and for the second novel nuclear subtype, quite interestingly, a lot of their marker genes seem to be involved in steroid signaling pathways. And in case of the third novel nuclear subtype, many of their marker genes were epigenetic regulators, such as polymer complex subunits. And finally, the last novel nuclear subtype, which we named the Rian-positive nuclei, mainly expressed a bunch of long non-coding RNAs, which are actually embedded in the same genomic locus as their time marker genes. So I just want to show some validation about the Rian-positive nuclei today, because I think this really shows the complexity of gene expression programs inside a muscle sync. So in case of the neuromuscular junction and myotendinous junction, they are associated with anatomically distinguishing locations. So their locations in the myofibers are always fixed in every myofibers that you look at. However, when we looked at Rian-positive nuclei, we could see that these nuclei were actually dispersely distributed throughout the myofiber without any clear positional preference. So for example, at times, these nuclei were located almost at the end part of the myofiber, sometimes on the middle. In some fibers, they were located quite far away from each other, whereas in some cases, they were quite close to each other. And the example of Rian-positive nuclei really show that these novel nuclear subtypes are not always associated with anatomically distinguishing locations, but there are more sophisticated mechanisms going on inside of myofiber. So lastly, in hope to identify more nuclear subpopulations, we also sorted out the bulk myonuclei from our previous data set, and we performed reclustering, which identified additional nuclear subtype that was not masked before. And when we tried to locate these nuclei by in situ hybridization, quite interestingly, we realized that every time we see these nuclei, they were always located at the very outer part of the entire muscle tissue. Here you can see this GFP positive muscle nuclei that are expressing the time marker gene mucin 13 and located at the very outer part of the entire muscle section. So at the very outer part of the muscle tissue, there is a specialized connective tissue layer called perimysium, which encapsulates the entire muscle tissue and stabilizes its structure. And EM study performed a long time ago have shown that there's a specialized adhesion structure between the outer myofibers and the adjacent perimysium layer, but this study has been overlooked afterwards and has not been really followed. But now we think that we might have identified the nuclear subtype that is responsible for establishing this kind of adhesion. So after successfully demonstrating nuclear heterogeneity in the normal state, we then asked whether we can see disruption of the normal heterogeneity in the disease state. So as a beginning point of this, we decided to first look at Duchenne muscular dystrophy, which is the most common muscle degeneration in humans, but yet it remains extremely devastating. So this disease is caused by mutation on a gene called dystrophin, and the normal function of the dystrophin protein is to link the actin cytoskeleton to the extracellular matrix of the myofibers. So if the cells lose dystrophin, then their membrane integrity gets compromised and myofibers undergo cell death. And what happens in the patient muscle is that the entire muscle becomes a mess mixed with various fiber stages, such as dying, intact, and actively regenerating due to the surviving stem cells. So we collected around 2,000 nuclei from 3-month-old dystrophy mutant mice, which are more commonly called as MDX mice, and performed single nucleus RNA sequencing analysis. And here you can immediately see that there's a huge alteration from the normal heterogeneity. And further analysis confirmed the notion that the MDX muscle is indeed a mixture of various fiber types And one way to see this is to calculate the gene signature score of individual MDX muscle nuclei based on the macrogenes that distinguish different time points of regeneration. And here you can see that the entire muscle became a mixture of intact and regenerating gene signatures. At the same time, we could see that a lot of these distinct nuclear cell populations that we found before were now gone in the MDX. And that even included nuclei at the NMJ. And quite interestingly, it has been shown that NMJ is histologically disrupted in MDX mice as well as in human patients. And probably our analysis is reflecting this aspect at the molecular level. But I nonetheless want to stress that this loss of nuclear subtypes was specific to some degree because other nuclear subtypes such as perimysium or sus2 positive nuclei were still intact and identified in this analysis too. But probably the most exciting finding from this analysis was that now we could identify several disease-specific nuclear subtypes with very well distinguishing markers. So today I will briefly talk about two of them, namely fiber repair population and dying fiber population. So in case of the dying fiber population, as you can see in this volcano plot, all of their marker genes were annotated to be non-coding RNAs, which you can probably guess from the gene names here. But when we try to detect these non-coding RNAs, we realize that these non-coding RNAs are interestingly marking the end-stage dying fibers, or in other words, necrotic myofibers. Now, because these necrotic myofibers have very leaky and unstable membrane, you can actually detect them with just using secondary antibody without any primary antibody. So in this image, you can see that the myofiber in the center is heavily stained with blue color, which is IgG, meaning that this fiber is dying. And you can see that this non-coding transcript is only very robustly expressed in this dying myofiber. Now, in case of the second novel nuclear subtype, the fiber repair, quite interestingly, most of their time marker genes of these nuclei were known to be directly mutated in various forms of human myopathies. Also, protein products of a lot of their marker genes were known to physically interact with one another, which are exemplified here in the below. And when we try to validate these findings by performing different combinations of in situ hybridization, we could readily identify these nuclei that are co-expressing these marker genes in the MDX mouse, but not in the wild type control. And this finding was further recapitulated in human patients who carry mutations in the dystrophin gene. So the obvious question is, what is the nature of these marker genes? In other words, what do they do such that they are so heavily implicated in muscle diseases? Now, because the skeletal muscle cells are generating huge force, their contractile machinery can be often damaged and ripped apart. So on the left side, you can see the transmission electron microscope of the normal muscle. And here you can see the beautiful organization of the sarcomere structures. Now, when the muscles are hyperloaded, some part of the sarcomeres can be damaged and become ripped apart. And it has been shown that the protein products of these marker genes are rapidly recruited to this damaged site and tries to repair this damage. Now, another type of possible damage on the myofiber is damage on the membrane. And once again, when the muscles are overloaded, some part of the membrane can be ripped apart and become open. And it has been shown that these marker genes are recruited to this site and tries to seal up this exposed membrane. Now, yet it is a bit unclear why this particular nuclear subtype expands in the MDX muscle. One possibility is that because their muscles are very weak, there is just high incidence of this type of damages. The second possibility is that it is just reflecting, recapitulating some developmental processes. But nonetheless, I think this has an important implication that the failure to properly repair this type of micro damages of the muscle is a common fundamental mechanism by which a lot of muscle diseases must occur. And it will be very important to understand how this repair processes is regulated, which will be instructed by the marker genes that I identified from this study. So finally, and lastly, we wanted to see whether modification of our approach can reveal nuclear heterogeneity in specific myofiber types. So as I mentioned earlier, there are several different myofibers which mainly differ based on their contraction speed and metabolic profiles. But we actually wanted to bring this task to the most challenging one and decided to look at muscle spindle fibers, which are specialized sensory fibers which regulate the muscle length and control our reflex regulation. So what makes the muscle spindle special is that in addition to the motor neurons, they are also innervated by sensory neurons, which start from the muscle spindle and project to the spinal cord. And by directly communicating with the spinal cord in both forward and reverse manner, the muscle spindle fibers play absolutely essential role in regulating our motor coordination. However, the problem with the muscle spindle fibers is that they are extremely rare. And therefore, in our previous analysis, we could not identify any nuclei that are coming from the muscle spindle fibers. So this is a schematic diagram of the muscle spindle fibers. I will not go into the detail today, but you can easily appreciate that it is a very uniquely structured myofiber. Now, the HSA Cree that I have been using so far do also label muscle spindle fibers. Now, although they are extremely rare due to their funny looking morphology, you can easily detect them in tissue sections, which are indicated in arrows here. But because these myofibers are so rare, if you randomly isolate nuclei from this muscle, then this spindle nuclei will be just diluted and probably lost. So we switched the Cree driver to carbine Cree, which was now able to recombine only in the muscle spindle fibers. And after isolating these nuclei, you're able to resolve clear nuclear heterogeneity within the muscle spindle fibers. And based on our own validation, and together with some known literatures, we are able to assign each of these clusters to different anatomical locations within the muscle spindle fibers. So to show you a couple of examples, on the top image, a gene called CACRL is expressed only from the middle part of this isolated spindle fiber, and at the same time, UFSP1, which is a marker of muscle spindle NMJ, is expressed as a distinct foci on the lateral side of the muscle spindle fiber. In the second example, once again, CACRL is expressed only from the middle part of this isolated spindle fiber, and simultaneously, piezo2 is expressed throughout the lateral part of this spindle fiber, but clearly excluded from the center part of the spindle fiber. So even in this rare and specialized myofibers, there are intracellular spaces and the transcriptomic programs are strictly compartmentalized. So how do all these diverse nuclear subtypes become established? This is still an open question and will be the focus of the future studies. But what I believe is that it is primarily regulated by different activities of different transcription factors. So when we profile the gene expression level of transcription factors and epigenetic regulators across our data set, you're able to see enrichment of different factors in different nuclear subtypes. Just to show you some validation that our data set is really trustworthy, ETV4 and ETV5, which were already known to induce some NMJ marker genes, were indeed enriched as the first and third rank transcription factors enriched in the neuromuscular junction nuclei. Another notable example was the enrichment of SMAD3 in the NTJA nuclei. And SMAD3 is a downstream effector of the TGF-beta pathway, which was already known to be involved in maintaining the myotendinous junction with mammals. But such effect of TGF-beta so far has been attributed only to their action on the tendon cells. And our data set suggests that the muscle cells will likely also perceive TGF-beta and use it in order to maintain the myotendinous junction. So to summarize my talk today, I have shown you that by single nucleus RNA sequencing, I was able to uncover previously unrecognized diversity and dynamics among muscle nuclei. And these different nuclear subtypes are usually located at anatomically distinguishing spots, such as the neuromuscular junction, myotendinous junction, or the perimysium. But this was not always the case. And as I have shown you with the example of Riam-positive nuclei, some of these nuclear subtypes are more dispersely located throughout the myofiber, without any clear positional preference. And also, as I have shown you, in the MDX analysis, some of these nuclear subtypes appear to be induced by local and acute signals such as damages on the myofiber. And collectively, these results show how regional transcription shapes the architecture of syncytial cells, and I believe that this will open up a lot of exciting possibilities in the future. So for those in the audience who are interested in the muscle, we also set up a web server called MyoExplorer, where you can freely navigate our data set. So if you're interested, please check it out. But if you're not interested in the muscle, then you can forget about everything that I talked about today, like the gene names, fiber types, or disease. You can forget all about them. But the real takeaway of my talk today is that conceptually, now you can consider syncytial cell itself as an analog of a multicellular tissues. And also these nuclei are contained in one cytoplasm. They are differentiated, just like differentiated cell types in multicellular tissue context. And I also want to briefly mention that my paper was published back-to-back with Douglas Milley's group in Cincinnati. And here what they did was to look at nuclear heterogeneity throughout the lifespan of the animal. And they were indeed able to identify specific nuclear subtypes during postnatal growth stage of the mice or in very aged mice. So clearly the dynamics of nuclear heterogeneity seems to, together with my MDX analysis, seems to play a very important role in regulating muscle pathophysiology. So this work was done done together with a number of really wonderful people, and I really would like to thank Carmen here for giving me unconditional support for this unusual project for the lab. And starting from February, I'm starting my own group at the IGBMC in Strasbourg, France. So if you're and our group will try to understand how these diverse nuclear subtypes become established and maintained inside shared cytoplasm, as well as their implications in diseases or during exercise and so on. So if you are interested, please feel free to reach out to me, or if you have anyone who might be potentially interested, please deliver the words. And with that, I will stop here and thank you very much for your attention. Thank you.